Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. We invite you into the stories, struggles, and breakthroughs shaping the future of human health. From cutting-edge brain science to discoveries transforming how we heal, adapt, and thrive. We explore the ideas that matter and the people behind them. Together, we look across diseases, technology, and research fields to understand not just what is changing, but why it matters for patients, families, and the future we all share. Join us as we uncover new insights, spark collaboration, and illuminate the science that can improve our lives. Welcome to Research Renaissance. I'm Deborah Westfall, your host and guide through the fascinating future of health science. Joining me today is Angarika Balakrishna, a PhD from the Brenhouse lab in Northeastern University, where she focuses on uncovering the neural mechanisms linking environmental exposures during critical early life development periods to long-term cognitive and emotional trajectories. So welcome, Angarika.
Thank you so much, Deborah, for having me. Really excited.
I'm thrilled to have you. You are, you've been doing some research that we've been, working with the Brenhouse lab. Heather Brenhouse has let you lead the discussions, which is just amazing. You being a big part of those projects. And I'm just kind of interested in really kind of getting to know you a little bit better. And let's discuss, how did you get into research? What was that moment or that influence that even took you down this pathway?
Yeah, so I think that's a very interesting question. And I think about it a lot because it didn't really start with a textbook or a lecture. It kind of just started at home. Both my parents are doctors, so medicine was always at the backdrop of my childhood. We had dinner conversations and they could veer into symptoms and diagnosis, and nobody would even bat an eye. It's like, oh, this is such a normal conversation to have at dinner. So I grew up in that world and I loved it. But I realized quite early on that I wasn't really interested in the clinical side, but I was really interested in the mechanism of what drives all of these clinical cases that my parents were talking about. Because my parents would ask, oh, like, what does this patient have? And I would find myself asking, like, oh, no, like, why did this patient have it? Like, at the levels of like the cells and molecules. And I feel like that distinction felt really important to me, even at such a young age when I was too young to even fully articulate it. And the real turning point came when I remember seeing this interview by my previous mentor, Dr. Vidita Vaidya, back at the Tata Institute of Fundamental Research. And she was describing this concept about, let's say, an early window. And she was saying that this is such a critical stage in a human's life, and it spans from the womb all up to like 12 years of age. And she said something that genuinely stopped me. She said that we have this strong feeling that this initial period is really critical in determining how your brain is going to be wired up for emotional responses in adulthood. And she really explained this very beautifully. And she said that there are many adverse effects that we, experience early on in life that have long-lasting consequences on the brain. And this is simply not the same as trauma that is experienced, let's say, in adulthood or even in early adulthood and old age. And what really struck with me was how she spoke about individuality. She said something around the lines of like, despite all of us sharing the same brain blueprint, we each develop such a unique emotional circuitry. And I remember this analogy that she had used. She said that, we want an architect that designs buildings and flats all in the same orientation, the same way, but then people populate those flats and they make it personal. They add their personal touch. There are like these different interior designs. So the same way the brain is built on this blueprint, we all have the same prefrontal cortex in the same place, yet there are still these micro level differences. And I guess after that interview, I was so fascinated by the brain and how it works, and I just wanted to understand more about it. I guess that's what really drew me to research and the kind of work I'm doing right now.
Yeah. So how old were you during this? Was this during college or?
I was 16. I
Oh,
was
wow.
at the age of 16, I was not even an adult yet. My prefrontal cortex was not yet developed. And I was just so fascinated by this. I was like, okay, yeah, this is what I want to do. Like, this is
Yeah.
my calling.
So from there, then high school, tell us a little bit about your academic journey. Where did you go to school? What were you kind of focusing on, and how did you get to the PhD candidacy here? So tell us about that.
Okay, so this is really exciting. So I sort of did so back home it's called the 11th and 12th grade. So I did my 11th and 12th grade with a major focus on science. So I think this was like conversations with my parents where I really felt that I did not still want to narrow myself down too much. I feel like there is so much that science has to offer, like the different systems in the body. If I started concentrating on just one system early on, I wouldn't be able to get the entire extent of it. So I had a very broad focus on science in 11th and 12th grade. And then I was sort of put on the conveyor. So in India, initially it was like, okay, it's either if you take up science, you become a doctor or an engineer. There is nothing in between. It's either black or white. There's no gray area. But my parents told me like, okay, if you want to like try out for the medical exam there, you can take it. But if you're not, that's not something you're interested in doing. You can take a general like science degree in college and figure out what you're really, really interested in doing. So I did sort of like go on the conveyor belt. I gave the medical exam, but even after I got a good score, I was just like, no, I don't think I want to do this. I did it to appease my parents, but moving forward, I started like really delving into science. I got a bachelor's degree in biotechnology. Again, a very broad field. I wanted to understand that whatever education I'm inculcating, can I take it from like an industrial level all the way to the bench? Like, what are all the possibilities that I can sort of scope out? So that was the purpose of doing my bachelor's degree. And then when it was time for me to enroll in a master's degree, COVID happened. So I was in a COVID batch and I was just like, oh, hey, I've always been the type of person who was just like, okay, go, go, go. Like, I just need to get to the next step. And I was just like, okay, which school is accepting master's students enrollments right now? And it was this university in Mumbai itself called NMIMS University. And I was like, okay, I'm enrolling in here. I just love learning. I want to learn more. So during my master's degree is when I sort of like started getting used to or getting sort of familiar with all the techniques that I know now, be it cellular biology, molecular biology, immunology, virology. So I got like a full plethora of knowledge there. And that is also where I went to do my thesis at the Tata Institute of Fundamental Research, where I met Dr. Vidita Vaidya. And during my master's thesis work, I got introduced more in depth to the field of neuroscience. And I went to her and I was like, I saw that interview that you gave in 2018, and I'm super excited to be working with you. And she was like, okay, that's fine. This is Prachi. She's my PhD student. You'll be working with her. And I was like, okay, great. So I started working with Prachi. And she's now Dr. Prachi Tiwari. She's a postdoc fellow at Johns Hopkins. And she introduced me to this wonderful field of psychedelics and like how even microdoses of psychedelics can have these anti-anxiety properties. And she was actually like looking at where exactly this particular psychedelic DOI acts on the brain and how does its action actually show up as behavioral outputs in rodent research. Also, I got ahead of myself. This is where I got introduced to rodent research and all the cool things we can really understand with using rodents as a model. And I guess that foray into research was like, okay, this is something that I could see myself doing for the rest of my life. And I stayed on in the lab for another 2 years as a research assistant. And I assisted on multiple projects of understanding how early life adversity affects the immune system, how early life adversity affects the metabolic system, how does it affect mitochondria, how does it contribute to like aging-related disorders. So I really, really enjoyed my time at the Vaidya lab, or as they call it, the Patment lab. And thanks to Vidita, she really motivated me to pursue a PhD to answer more of the fundamental questions that I'm hoping to answer. And fast forward to right now, it's been I joined in 2023. So I joined the PhD program in 2023. I've been here for 3 years and I'm still uncovering this beautiful organ, the brain, and how early life adversity affects the underlying neural circuits, how it affects the endocrine output of the animal. And again, I'm really, really happy that I got that broad scientific knowledge when I did, because right now I'd be like, okay, I'm just focusing on the brain. But no, the brain is the master of organ, which means it controls further organs in your body. And I realize it's really important for us to be looking at all the interactions, not just the brain.
Is that area of study, is that pretty broad, or they're just is it pretty niche, looking at that early development and the journey across the life of an individual? Because a lot of times we think about the brain and the disease as just what happens when you age. The brain's there, it's working, yes, there are things that people are born with, but, that idea that maybe something that happens younger, which doesn't really show up maybe till you're older, is that an area that's emerging, or has it always been there and it's more of a niche?
I think that's a very interesting question. One also, I'll give a little shout out. So I recently published a paper that I'm really talking about this developmental period that is sensitive to traumatic events and how does that sort of shape later life behavior. But to come back to your question, I think people consider it to be niche because people think that early life adversity is a monolithic event, because we use early life stress as though it's a single variable that we can just dial up or down. But when you actually go into the biology of it, you realize that the timing of an adverse experience and the type of adversity, whether it was predictable or random, whether there was a safe relationship in the middle of it, it all produces very different biological signatures in the developing brain. And I think this is an area that we should really be focusing on, because even within the perinatal period, this period that we're talking about when the brain is developing and all these changes are happening, because a child that experiences chronic neglect in the first year of life looks neurobiologically very different from a child that experiences one acute trauma at, let's say, age of 8 or 9. And these stress response systems are implicated in different epigenetic changes are different, the behavioral outcomes are different. And then when we sort of collapse all of this into one single construct that's called adversity, we lose the mechanistic precision that would otherwise allow us to really target these interventions effectively. I like to say that we've been treating adversity like a dial when it's more like an instrument panel, and there are 20 different switches, and even a flip of one switch can produce dramatically different experiences. So I really think that while this field is very niche, there are many avenues that we can expand upon. And like I said, within the instrument panel, really studying each and every switch is really important, because right now, the only thing we have established is that adversity is bad for the brain. And we really don't know which kind of adversity, at which moment, in which biological systems. And I think that's where the actionable science lives. We've been kind of slow to get there, but I think we're getting there bit by bit.
So define adversity. And I know we've had this discussion before in other venues about, because some would say, well, one person's adversity is just a normal day for another person. Can you shed some light on why this is important, and it's not necessarily just a feeling, it's a serious kind of condition, if you would, or
Yeah.
I may not be defining that correctly.
No, I think this is a question that I have been thinking about a lot. I keep thinking about okay, I see, let's say, two siblings. They are experiencing the same sort of chaos at home, but one of them emerges resilient and the other one is just stuck there, sort of as we call broken, and they don't know how to function normally, or that comes out as behavioral outcomes or even certain genetic outcomes that happen later on in life. So I think because we treat like I spoke about the group blueprint, like we all are built on the same nervous system blueprint. What we fail to understand is that there are many other underlying circuits and cells that change so differently in different people that it's really hard to pinpoint and say that, okay, you let's say, for example, since I study maternal separation, you did not have a mother from years 5 to 10 growing up, and you're saying this to two siblings, and one of them was like, yeah, well, somebody had to step up and look after the household. Like, both of us couldn't just, like, sit back. That also comes into play that, okay, what circuits are got rewired in each sibling's brain that has caused them to act so differently or react so differently to this adversity that we see. So usually in the lab, when we define adversity, it's just like any negative experiences that a child experiences early in life. This could be from the day of birth all the way up to the ages of 12 and 13. But what we fail to recognize is that a negative experience to one is not always construed as a negative experience to another person. So again, it is very subjective in humans. I can answer it on the fundamental level. In rodents, we just see the genetic changes, we just see the neural changes, we just see the behavioral changes. But that's because we cannot communicate with rodents. The communication bias comes in. Some people are better able to express what they felt, some people are not. And I guess there's just this lack of standardized tests to really determine that, okay, what can we really classify as an adversity? There's a scale of adversity. Where do you lie on the scale of adversity? So I guess all of this is just something that we lack in terms of standardized procedures to come at. And I think with a little more research, with the help of like other models, not just rodents, let's say chimpanzees, you have other Drosophila and all the other model organisms, if we can answer that at the fundamental level, I think it's easier to get to it at the translational level in the future, hopefully.
Mm-hmm. So today you are studying, you mentioned maternal separation, and you're in Dr. Heather Brenhouse's lab up there in Northeastern. Tell us a little bit about your focus. What is it that you're studying? I think you're using rodents, but what are some of the hypotheses or the questions that you're trying to answer?
So in one short sentence, what I'm really interested in looking at is how does this early life adversity, in the form of maternal separation, affect the development of this neuroendocrine system? So I guess it's like I want to get through the neck of the river before it floods, right? So I want to really understand what is happening in the brains during development that's really shaping future trajectories. So some of the hypotheses that I'm trying to tackle is that we're all very familiar with the hypothalamic pituitary adrenal axis, which is our stress response axis. And while we look at the axis as a whole, we see that, okay, at the end there's a cortisol release, and that is our stress response. But what I really want to understand what is happening in the seat of the axis, what is happening in the hypothalamus at the molecular level that is driving the stress response. And are there any targets within the hypothalamic region that can be used to treat anxiety-like behaviors, depression-like behaviors that since years people have been trying to sort of get a treatment for? And one of the hypotheses that I've formed is that there is this receptor in the hypothalamus that is the CRHL1 receptor that is very crucial for the functioning of the HPA axis. And I have found that if we target this receptor early on and we prevent the downstream effects of this receptor, we're able to prevent the downstream effects that early life adversity has on the body in the form of early onset of puberty. It's not news that girls, especially girls, boys too, but especially girls are hitting puberty earlier than normal. But nobody understands why. But we know that early life adversity is a contributing factor to early onset of puberty. And I found that in rodent models, if we target the CRHL1 receptor, we're able to prevent early onset of puberty. And what we're also able to prevent is the increased anxiety-like behavior that we see in adolescents. Now, again, adolescence is this period that our lab is really interested in understanding and in sort of targeting, seeing that during this developmental period, if we target the adolescent time points for various diseases, are we able to get any positive outcomes in adults? So one of the hypotheses that I'm now working on of late is that we all know that exercise is good for the body, but is there benefit in sort of exercising at particular time points in life over others? Because obviously you can exercise whenever if you want to remain healthy, if you want to tone up your body. People exercise for just endorphin release. They actually get as a high, as they say, when they exercise. So I'm really interested in understanding if we administer exercise as, yes, like that, administer exercise as a treatment paradigm during this adolescent period. Are we going to be able to prevent any of the downstream effects that we see in terms of metabolic changes? Like, do our mitochondria, because we know mitochondria is a powerhouse of the cell, do our mitochondria function better? The mitochondria affect all the cells in the body, the neurons, the glia that are like, our immune cells, microglia. So can they affect metabolism? That means, can it affect the immune system? Can it affect our nervous system? Can it affect our brain? Can it prevent aging-related deformities that we see in the brain? Can it prevent aging-related cognitive declines that we see in the brain? So this is something of late that I'm really interested in understanding. And also, people say that the reason for early onset of puberty, more often than not, is polycystic ovarian syndrome in females. So, and a lot of doctors, endocrinologists, prescribe exercise as a treatment option to sort of lose weight and also lose the negative effects that PCOS has on the body. So I'm like, okay, if I'm saying early life adversity causes this early onset of puberty, if I tell the patients, or in my case, rats, okay, just exercise, you will not see this early onset of puberty. Neither will you see metabolic changes, immune changes, and cognitive declines associated with aging. So that is something that I'm really trying to get at. And the last one I get to...
Can I ask, what is the downside of early onset puberty? I mean, that's just not something I think people that have children worry about. And when is that? Like, is that the age of 8? Is it the age of 9? Is it 13, 14? And why is that a bad thing?
So typically, I'm going to talk about females more often than not, because I think, again, a very important topic in terms of rodent research or fundamental research is that we don't really study sex differences, and we don't realize that all this while we've just been studying male models, and we really need to focus on the female model too, because again, our biology is so different. Mm-hmm. So in terms of females, typically females are sort of on track to hit puberty at ages of 12 or 13. But now, because of changes in the environment, changes in caregiver sort of presence, that has affected female pubertal development, and it caused them to have puberty as early as ages of 8 and 9. Now, the negative consequences of that are both immediate as well as long-lasting. Immediate, they don't know how to react. They're like, okay, like, nobody told me to be prepared for this at the age of 8 or 9. I don't know what to do. Do I talk to other people in my class about it? Will they think I'm weird? Have they even heard about it? So that comes with like just a mental health toll of even navigating that entire system when you're not even used to hearing about it yet. You've not had your parents tell you about it. Nothing. Secondly, the body usually develops in such a way that it is able to handle insults at a certain age. For example, let's say menopause, right? Like, nobody expects to hit menopause at the age of 20. So your body has not developed at the age of 20 to actually withstand, let's say, the hot flashes that you get from menopause or any of the other behavioral outcomes that are associated with menopause. So that's a toll on your physical health of your body as well. That, okay, I am hitting puberty at a much earlier age. There's this surge of hormones in my body that is going to have mental outcomes as well as physical outcomes. Now, that is the more like acute or immediate effects that you can sort of see with early onset of puberty. With the long-lasting effects, it's more on what changes we see when we move this critical period of development earlier than normal. Has your brain had enough time to sort of form the connections that it would typically form if it didn't have to sort of rewire itself and be like, okay, now we have to produce all these different hormones because pubertal development has started, so we need to, sort of dedicate all of our resources to that instead of dedicating the resources to the actual formation of neural circuits that could be controlling, let's say, anxiety related behavior. Now, this is all speculation. We have no sort of like concrete evidence that the brain is rewiring itself to produce hormones when it could have been rewiring itself to regulate anxiety related behaviors. But there is research that shows that females who hit puberty earlier than the typical age range have increased anxiety like behavior later on in adulthood. They have depression related behavior. It sort of makes them vulnerable to a whole host of mental health disorders. More often than not, it's also linked to obesity in females. And we know obesity in turn, again, affects different organs in the body, affects your immune system, your endocrine system. More often than not, females suffer from the polycystic ovarian syndrome that I was talking about. It has led to infertility in a lot of females are not able to get pregnant. And while at that point, our endocrinologist can just be like, okay, like, no, we don't have the hormones that are required for pregnancy, but we don't know exactly when did our body stop or our brain stop rewiring itself or telling us that, okay, no, you're going to stop producing this hormone now and you're not going to be able to have kids 10 years down the line, 20 years down the line.
I see. Yeah.
So that's important to like pinpoint that leads, that developmental behavior.
Yeah. That's really interesting. So, you mentioned some of the questions around the development, around exercise, what is happening, not only inside the brain, but kind of the emotional side of the brain. What are some of the questions that you think, maybe even just the most important question that you think needs to be answered right now in this area?
I guess it's just more than a question. I think in order to formulate proper questions, we need to stop treating adversity as a monolithic event. But we need to start asking the questions of like, okay, I have 20 different reasons that early life adversity could be bad for you later on in life. Which reason do I want to target? Or do I want to look at all 20 reasons? According to me, I think we should be looking at all 20 reasons, and we should really be very, very thorough when we are sort of trying to devise treatment options for early life adversity later on in life. And if we keep treating adversity as one single event, as a single dial, we're never going to be able to get to that. So I really think that we need to be more thorough with the questions we ask. And it can't be a single question. It has to be several different questions that need to be asked. Because, again, if, let's say, a single person has question number 3 that comes up in their analysis of like why early life adversity could be bad for them, another person could have question number 19 coming up, and we will never know if we don't go through the entire set of the 20 questions. So it is a lot of hard work, but I really think we have to go through the entire array of different questions.
Yeah. Well, I think what you're bringing up, which I think is fascinating, because I've interviewed others that are in the mental health side of depression, mental health, anxiety, is this may really be a biological issue in some. It's not, for so many years, and, I would say in so many families or so many cultures, it's feelings, right? It's feelings, just get over it, make yourself happy, go walk in the sunshine and you'll get over it. But I think what you're saying is, there are environmental things that are creating the adversity, but it's translating to biological changes in our bodies that are not just feelings for, buck it up, we've all had a rough day. Can you expand on that?
It sort of narrows down to if we treat biological variables and environmental variables as two separate things or one same thing. And I think this question is really like holding the field back, and it's a fairly strong question because we often use biological and environmental variables interchangeably. And while right now, like in the olden days, we didn't have the tools to really parse apart what's biology versus what's environmental. And I think that right now, even when we have the tools and we're able to separate separate two of them as biological and environmental, it seems somewhat artificial, really. And it all narrows down to we know that the environment shapes gene expression, and we know that early life adversity can sort of alter this gene expression and can determine when genes are switched on, when they're switched off, and this could persist across decades. And we also know that genes shape how the environments are actually lived in or how they're experienced. Because your genetic makeup influences the kinds of environments you're drawn to, the way you interpret social cues, how your body sort of responds to its stress. And all I want to say that in all of this, the brain is not a passive screen on which the environment simply just like writes or like dictates its sort of different cues. And the brain or biology is actually an active participant in constructing the experience. And I think the reason that this distinction still persists is partially because of scientific inertia, but partially it's also political. And the reason I say that is because biological is coded as fixed, like this is a fixed determinant, we know it exists. And we think the environment is changeable. And the framing has real consequences for how we fund research and how we design policy. Because I think both the premises are wrong. Genes are not the end all be all. They are changeable, they are plastic, and environments are harder to change than you assume. For example, somebody going through early life adversity, it is easier said than done that just take yourself out of this adverse condition and things will be okay. But no, you cannot. Like environments are not that easily changed. And which is why I think that we really need a new framework that, holds this reciprocity honestly. And it does not fall into, oh, everything is connected. Biology is connected to the environment. The environment does not change. Biology changes. No, it's not interchangeable. Genes change. Neurons are plastic. New connections are formed all the time. The environment is not as easily changeable. And I think that's something that we really need to account for in our experiments when we design scientific projects, and we really need to tackle them as two separate actionable items. And I think that's when we really get to the core of, okay, was that environmental changes that caused the changes in the brain, or was it changes in the brain that caused us to perceive the environment differently than we would have?
Yeah.
No.
Yeah, and I think I go back to this, this maternal separation question, which is not you didn't have a mother, you just may not have had a mother that gave you care. You may have had a mother, but there wasn't that loving, caring, meet your needs as you developed. I'm really curious, do you have these conversations now with your parents? Because you said they were, what is happening? And you were asking why this is happening. Are there debates around the kitchen table now when they're, how does this work? Because you're asking the biological, the environmental, is this shedding light on what they do? Like, I'm just really curious how this works.
I think so. Right now, my conversations with them are pretty limited. We have a 10.5 hour time difference. So when I call them, they're just like, okay, are you okay? I'm like, yeah, I'm okay. And I ask them, are you okay? They're like, yeah, we're okay. And that's the end of it. So I sort of don't get much time to talk to them about this, but the few times that I've actually talked to them about it, my parents are also very curious about the fundamental research that I'm doing in the laboratory. And when I'm explaining to them, they're just like, okay, but like, how does this sort of like translate? Like, okay, you're doing all of these cool things in animals and you're finding out all of these cool scientific discoveries, you're getting like treatable targets and stuff like that. But how do you anticipate like translating this to humans? And I think that, again, I don't want to have a rushed answer for it because I think that anything that's rushed, even like rushed into translation, people have always paid a price for it. You cannot rush into anything because the mechanism is not exactly the meaning. Like, okay, we found this cool mechanism, but that's not the end all be all. For example, like when I found a change in a particular circuit in a rat that has undergone like early life adversity in the form of maternal separation, what I have is a biological fact. And that's fundamental. That's all I have. I have a biological fact. And that fact is real and it's really fascinating and it's important, but it only becomes useful when I can connect it step by step to fascinating human being experiences and to something that can actually be tinged. And that's why like my parents have put me in touch with a lot of like neuroscientists back home, and they're all like at the clinical realm, not just as a courtesy, but they really talk to me because they're generally interested in research that I'm doing. And it really helps me not constrain my thinking in a certain way. It's just like when my models are too clean, when I have these 4-star significance, I push back a little. I'm like, wait, hold up. That can't be real. Like, I'm seeing all these cool effects, but it's not all black and white in humans. There's so much gray area. There's so much else to consider. And in rodents, again, it's a biological output. It is what I see. There is no other output that I could have. But with humans, like lived experiences change a lot. For example, let's say that a patient is undergoing cancer treatment, right? And there's one patient that has family support. They're going through it with like 4 other people in the room at all times. They're never alone for a chemotherapy session. But there's another human getting the same exact job, same dose, same amount of times, but there's nobody with them. And I think that really changes how the outcomes of the patient are, because it's, again, it's social. We are social beings as humans. And I think social interaction is so important to us. That will, again, in turn, rewire our brain so differently that we will have different, hopefully positive outcomes for both, but we will have different positive outcomes. So in that sense, with these conversations, I try to sort of like include the perspectives of people with lived experiences of the things that I actually study. I can talk to people who have had some trauma early on in life. I had my childhood best friend go through some sort of trauma, and I talked to her about it. I try to explain to her, like, okay, like these things have changed some sort of thing in your brain, in your body, and this is why you're seeing any of the health issues that you have right now. And I also see these gaps in a mechanism that look invisible in the lab. Like I was like, okay, this is not a problem in the lab because I'm studying rodents, but these gaps are there in human research. And I think that the conversations evolving around translation and just understanding, talking to people with these lived experiences is really important. And I'm glad to have people that I can have these conversations with.
Yeah. It's interesting as you were talking about the just the scientific method, right? There's so many people that, well, why don't we have a cure? Why don't we know, right? It is that behavior of a scientist, which you just described, which is if I have a fact, I can't translate that without other facts. If I have something that's very new and stellar, I think you called it like a 4-star, I need to question why versus just take it as a fact. And that takes a long time. And, how long have you been going to school since you got out of high school, 11th and 12th grade?
So ever since, I feel like
years, right? It's like
years. Like even when I was not in school, I was constantly like learning about things, just like different things that happen around me, different advances in neuroscience.
But it's been 12 years and is it 12 years now?
So
10?
when I got got out of high school, I was 16. Yeah. Yeah.
Yeah.
I'm 27. It's been 11 years.
And
That's
you are
crazy.
you're going to get your PhD. When do you defend?
I hope to defend next year, December, because I sort of came in with my master's. So I'm trying to finish my PhD in 4 instead of 5 years. But if I'm able
12
to get it,
years. 12 years to get your PhD, and then your career just kind of starts because now
that's
you right now you've got your PhD, and now you have to go through and do all the other things that you need to do. And I think that is what's really fascinating to me is how, first of all, the dedication and the time, but how long it really takes for a person like you to go through all the training, the formal training, and then collect all these experiences that you get to really now start to study the specifics and, to come up with some of these, some of these breakthroughs. Tell us about your paper. You mentioned it. You gave it, what is the name of your paper and what was it published? Because this is a big deal. It's a really big deal, especially for a PhD candidate. Tell us about it.
so. Firstly, just quickly, I think even after my 12 years, I'm still going to be constantly learning. Like this
Oh, for
field
sure.
is evolving, it's
Oh, yes.
changing. I'm constantly going to be learning. I don't anticipate stopping learning until like I'm retired. I'm like sitting at like a beach somewhere, like sipping, and until then I'll be thinking like, God, like my brain is rewiring right now as well. But anyway,
It won't shut off. It won't shut off. I
it
promise
will
you that.
not.
Yeah.
Um, so I I actually have two papers that recently got published in the past 2 weeks or so. So one of them is where I'm actually talking about the CRHR1 receptor, and I'm targeting the CRHR1 receptor and seeing how that prevents early onset puberty as well as anxiety related behaviors in rodents in adolescents. And that has been published in Hormones and Behavior. And the second paper is where I'm talking about where, like, okay, we do this maternal separation paradigm for a period of 20 days. But again, like I said, like, not everybody experiences the maternal separation paradigm or the maternal separation or even adversity during the same period. It could be directly after birth. It could be a couple of days. It could be a couple of days after birth, a couple of months, years. So that was a question that I was tackling with my second paper where I'm looking at different sensitive windows in the rodent model during which adversity could have different outcomes in adulthood and in adolescence in terms of behavioral outputs, in terms of endocrine outputs. And now, even though this work is not published, I'm also looking at the neural outputs that it has in these animals. And this paper has been published in the neurobiology of stress. And I do want to say that none of these publications and none of this work would have been possible without the generous, generous input from the Parent Health Charitable Trust. Like, I am so grateful to you guys, especially given the current economy. You guys are still believing in the science that we're doing. And it's really, really heartwarming. And I'm really grateful to you guys for that.
Yeah. Well, you're welcome on that. And we appreciate the plug too, because, this, having people like you early in your career, we've got to keep the pipeline of researchers. There's so many things out there that we don't know about our bodies, our brains, the diseases. We need answers, and if we don't keep the pipeline filled, then we'll lose that that opportunity there. So no, we appreciate you too. So. When you kind of look out in the next 10 or 15 years, you're having, the same conversation. What do you hope to really have fundamentally changed in this area of research? And it may be around kind of mental health, or it may be stopping some of it, the biological impact. What do you hope for?
That's a very interesting question. And I think this is going to be sort of like anticlimactic to what I am hoping to do right now. But I think that my hope is that 10 years, 15 years down the line, we will have moved past diagnosis as the death. Because the system that we have now is that, someone receives a label that implies a categorical disorder with a corresponding treatment. And it's increasingly out of step with the biology that it actually shows us that mental health disorders are so dimensional and they shift across the lifespan. And we have to remember that all these mental health disorders, they have shared underlying mechanisms across what we currently treat as separate diagnostic boxes. For example, depression, anxiety, PTSD, certainly personality disorders, even they share the same circuitry, they share similar inflammatory pathways, and they also share similar stress response signatures. And I think the mistake that we're doing is in treating them as fundamentally different conditions. And this has really slowed us down. And I really hope that in the future, a couple of years down the line, we're doing more trans-diagnostic work and we're targeting specific neurobiological processes that cut across all of these conditions. It's like, as the grad students in my lab like to say, like feeding two birds with one scone, right? Like we could look at all the emotional regulation circuits. We could look at the inflammatory clone. We could look at the stress reactivity. And I really hope that we've moved from the intervention point upstream, like more towards the developmental trajectory and in real time, not just rhetorically. And this is the goal that I sort of carry with me. And I mentioned this earlier, that it's catching the river before it actually floods. And a world where a child's developing brain gets the conditions it needs, the safety, the relational warmth, the sensory stability, before the absence of those things becomes a person's defining strengths. And I think that's my 10-year goal. And I think it requires all neuroscience, policy, culture, all of that sort of like moving in tandem. And I think it's possible. I have to think it's possible. That's what I'm working towards.
Right. I think that's great. When you were saying these boxes, right? I was just, and this might be just really out there. I wonder if we really get to that point, what you've talked about, if we really realize that we're all more the same, then there are these categories of people that have these conditions. Because if you don't put it in a box, when you say mental health, that opens it up for a lifetime of experience where maybe one individual has struggles a little bit at a certain time versus not, and it doesn't categorize. Does that make sense?
No, it definitely does make sense. But I think, again, I know this is like just additional work for people to do, but I think this work is important. Like, if you don't categorize it, like you said, it sort of opens up Pandora's box. Then it's like, okay, like a multitude of things could be wrong. Like if we categorize it, it helps us sort of like target the disease. But no, it actually does not. If you categorize it, it does not. You have to treat it as something that affects the entire body. It's not just a mental health disorder. It's a physical disorder. It's an emotional disorder. It affects every part of your body. You have to get better at not categorizing it and treating it as a whole, as everything that could potentially be wrong, go wrong, or affect things in the future.
Well, that becomes very personalized to the person then.
It's true. It does become personalized. But I think that's why it is important to have all these huge databases and repositories that we actually have. And we should be good about feeding data into those repositories. I think that's when it becomes important to have open access science. So we're actually able to gain all the knowledge from around the world, people's different lived experiences. So we have all these data points to actually make a conclusive search or research activity.
Well, unfortunately, we are coming to the end of our time here, and it's been wonderful. What do you wish people knew about what you do that they probably don't have any idea?
In terms of just my work, or is it more like as a researcher what I do?
Yeah, as a researcher.
So there's this like archaic academic construct, right, that we have that all researchers sort of just, it's like an unsaid rule. All of us just like follow it. And I feel like science rewards certainty more than it rewards doubt. And, it's always like you present your work as though you've always known where it was going, and you frame your negative results as, oh my God, like we didn't learn anything. But, we didn't get what we were hoping to learn, but this was informative. And you perform a kind of like narrative coherence of like your research trajectory. And it's often very little in relation to what actually happened. And what happens with young researchers such as myself is that we absorb their performance. We see keynote speakers, putting up their performance all the time. And I feel like in doing so, in sort of absorbing that and trying to replicate it, we become slightly worse scientists, because I feel like we sort of lose our authenticity in that way. And I really feel like that is something that we need to change. And that is something that I'm trying to change from my work too. Like the sort of imposter syndrome that we are all suffering from. I try my best not to suffer from that. And like, I really see Heather doing that in lab meetings as well. And it's sort of this honesty in practice where I see Heather just getting up in lab meetings and be like, okay, I don't know. Like, I don't know why that happened. And we actually share our failed experiments as openly as we share our public citizens. And we see confusion as a sign of engagement rather than a sign of incompetence. And this is something that's really helping me grow as a scientist. And I'm grateful to Heather and everybody else in the laboratory that I work with for that. And I really feel that the mental health crisis that we all are experiencing right now, and in graduate training specifically, is real. And it's not being met with anything close to the seriousness that it deserves. And we have to realize that the data is not ambiguous, that the anxiety and depression rates among PhD students are drastically elevated. And we are losing people in the field who came in very curious and capable, and we're not losing them because they were not good enough, but it's because the structure is what brought them down. And this really should be alarming to everybody who cares about the long-term health of this field. And I really think that is something that I'm trying to practice with my research as well, taking the breaks when needed, answering the fundamental questions, getting it from all different avenues. Failure is just a part of the process. And I'm really grateful for Heather for supporting me. And I think something that I would really like to tell everybody to do is don't hold your results to your heart. Hold them more lightly and interrogate your your methods more aggressively than interrogating your results. And I become more interested in my errors and troubleshooting my errors than I have in my successes. It's like, oh, when an experiment fails, oh my God, this gives me an opportunity to troubleshoot something. So I'm really just taking it as the day comes and trying to better my science, make it more accessible to people, communicate my science better. And I think, yeah, that's all.
I think that's wonderful, and I think that's a great place to land. So I really appreciate you spending the time today. I know you're very busy. Where will people get a hold of you or find your research? Do you have an email? How do they get a hold of you?
I have an email. I can sort of you can link it to the video.
We'll put it in the show notes, and but you'd be happy to hear from them?
Anytime. You can email me anytime. I don't have any work hours. I email outside, inside. I obviously do not expect you to respond to me outside of work hours, but I am always happy to get questions, answer questions. You can also find my work on the Brenhouse Lab site or the Northeastern site. And I'm in the process of starting developing my own Google site. So that should also be available soon. And I can share it with Deborah. And you can link that to the video as well.
That's wonderful. That's wonderful. There's people out there that are listening that would like to find out more about what you do or possibly even to fund your work. So we want to make sure that they can get in contact with you. This has been fabulous. Thank you so much. And
thank you.
Yeah, we will talk very soon.
Talk soon. Yeah, thank you.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.